Most people have heard of DNA. Good old deoxyribonucleic acid. But what do we get from DNA? Why is it so important? As it turns out, the information held in DNA codes for proteins that carry out many important functions in the body, like aiding in the immune response. Today, we will discuss how these proteins gain their shape, a process known as protein folding. These 20 substances in the chart, known as amino acids, make up all of the proteins in the human body. Each of them share a similar base in yellow and have a unique R side chain, which is the part highlighted in blue. The primary structure of a protein consists of the order of these amino acids, coded for by the sequence of DNA. Secondary structure is the folding of these strands of amino acids into either alpha helices or beta pleated sheets through hydrogen bonding. Tertiary structure, the final level of structure for many proteins, is arguably the most confusing, formed using all these types of attraction that depend on the properties of these 20 complex amino acid R side chains. I find it much easier to think of this process through the frame of dating. No, not carbon dating. Dating. For instance, imagine that these two amino acids signed up for an online dating service and, through all the craziness, found someone with similar interests and dislikes as them. Here, both glycine and valine, being hydrophobic or literally afraid of water, would for sure swipe right, feeling that mutual attraction. In this case, like attracts like. Since the majority of the cell is in an aqueous environment, meaning it is underwater, hydrophobic amino acids like these two often gather on the inside of the protein to shield themselves from the water that they fear so much, leading to these hydrophobic interactions. Other R groups can form hydrogen bonds if they are so compatible, which is another type of interaction you'll see in the protein tertiary structure. In ionic bonds, too, opposites attract, the negative ion being attracted to the positive ion. Finally, the amino acid cysteine can combine with another cysteine to create a covalent bond known as the disulfide bond, making this the strongest interaction in the protein tertiary structure. All of these interactions work together to create a folded protein that carries out a specific function in the body. However, if the temperature or pH strays too far from that protein's ideal range, it undergoes a process known as denaturation, in which the protein unfolds because its bond breaks under those conditions. That is why eggs solidify and change color when they are fried. I guess that when times get rough, sometimes relationships just don't work out. It's sad, but it's true. The disulfide bond, though, is the strongest interaction, making proteins with more cysteine amino acids more resistant to denaturation. Talk about a stable relationship. In all seriousness, though, the tertiary structure of proteins is very important because misfolded proteins often cannot carry out their intended function. Misfolded proteins appear to cause many diseases such as cystic fibrosis, Alzheimer's, and Parkinson's. Cancers, too, can be caused when the sequence of a certain cancer-suppressing gene is changed, decreasing the function of the resulting protein and allowing for the unchecked cell growth that is characteristic of many cancers. In conclusion, the attractions found between different types of amino acid R side groups is what allows for the protein to fold and function as intended, leading to life as we know it. Love. That's what makes a protein a protein.